everyone. Welcome to OsteoBytes. Thanks so much for joining us today. My name is Christina Iptoma, and I am mom to Osteo Angel Dylan and Director of Scientific Programs at MIB Agents. And today on OsteoBytes, we are joined by Dr. Shahana Mahajan, one of our 2024 OutSmarting grant recipients, and she will provide an overview of her funded work on repurposing drugs for metastatic osteosarcoma, specifically with a drug called Riluzole. Thank you so much, Dr. Mahajan, for joining us on OsteoBytes today. We are thrilled to have you. And thank you also to our panelists, Walker and Sona, for joining us. Everyone, please feel free to submit questions during the presentation through the Q&A in Zoom, and we will share those with Dr. Mahajan after her presentation. A little bit more about our guest today. Dr. Mahajan is a professor and principal investigator from Hunter College of the City University of New York, and she earned a PhD from the Indian Institute of Science, a premier institute in India in the field of molecular biology. She did postdoctoral studies at Weill Cornell Medical College in the Department of Pharmacology and at NYU Langone Medical Center in the Department of Biochemistry. And she continued her research at NYU Langone Medical Center as a research assistant professor until she joined Hunter College as an assistant professor in 2007. At Hunter, she continued her research in neuroscience to investigate glutamate-induced excitotoxicity in rat hippocampal neurons. And after her lab was lost to Hurricane Sandy, she reinitiated her research in osteosarcoma. So that was one good outcome, right, of Hurricane Sandy. In 2015, her lab moved to Belfer Research Building, which is part of Weill Cornell Medical College, and she was appointed as an adjunct faculty at Weill Cornell. Thank you so much for joining us today, Dr. Mahajan. We're really looking forward to your talk. And I have to admit, when I first came across the drug name, it looked like some delicious new pasta variety. I was like, oh, it looks like, Riluzole and like a delicious bolognese sauce. But now we know it's pronounced Riluzole. So really looking forward to your talk. Just a few announcements before we get started. It is Halloween, so happy Halloween. If you want to give yourself a Halloween treat today, I suggest listening to the latest episode of our AYA Osteo podcast. It's where words spill the tea on living with osteosarcoma as a young adult. And the October episode is perfect for today. It's called Spooky Sarcoma Stories. And Mia, Sammy, our co-hosts, are joined by Jillian, Mackenzie, and Matthew, and they share spooky stories from their osteosarcoma treatment experiences. You will cringe and cackle at the haunting stories of pain meds gone wrong, from dreams filled with housework and little men constantly appearing in the hospital room, and also mysterious screams behind a curtain. So really funny stories. Maybe listen to it on your commute home today. I think you will really get a kick out of it. And I'm sure Walker and Sona, you guys can probably relate to some of those stories. Also save the date. Factor 2025 is going to be June 26th to 28th at the Grand America Hotel in Salt Lake City. Registration is going to open in early February, but we are already accepting abstracts. Those are due on January 10th, and I'll put a link in the chat for more information about that. Next week, we also have another Turbo meeting. Turbo is our virtual tumor review board for osteosarcoma. It's an educational forum where clinicians discuss complex cases. It's also a great way for researchers who don't normally see patients to learn about real-world treatment challenges. It is open to medical professionals only, but if you are a patient, you are welcome to ask your oncologist to present your case on your behalf. Our next meeting is next week on November 6th. 5 p.m. Eastern. If you would like to present a case or just interested in attending, you can email me and I'll put more info in the chat about that. Lastly, today's Halloween, but tomorrow it's November 1st, and that means it's the first day of Pet Cancer Awareness Month. So you can support our Outbarking Osteosarcoma fundraiser and order a fashion statement bandana for your doggy, which I have here, which weirdly feels like Italian silk, though I know it's really not, but it feels like really soft and silky. So you can order one of those and take a picture of your pet wearing their bandana and enter the photo contest. It's super fun. I'll put a link in the chat where you can get more information about the bandana and the photo contest. And that is it for me. Let's get on with the presentation. Sona, could you please introduce yourself?
Sure. Hi, everyone. My name is Sona. I am currently a senior at Tufts University, so I'm calling in from Boston, and I am a pre-medical student studying biology. And I'm also an osteosarcoma survivor. I'm about 4 years NED. And with that, I'll pass it on to Walker.
Yeah, thanks. Hi, my name's Walker. I'm part of the Series Junior Advisory Board and have been for the last few years. Like Sona, I'm a senior in college. I'm at the University of Kentucky here in Lexington. I'm really excited to hear more about today's topic. I got a sneak peek with the slides yesterday, but Dr. Mahajan, you can take it from there.
Thank you. Thank you, Christina, for the introduction and for inviting me to give this talk. I'm so thrilled. And thank you, Sona and Walker. I'm going to share my slides. Yeah. So Christina actually introduced me very well. Thank you. So this is my brief history in pictures here. So I started in 2007. Only thing that I want to add is that the osteosarcoma work we started doing, we started around the time we moved to Weill Cornell. And in 2020, I became a faculty at the Graduate Center, CUNY Graduate Center, which means that I could actually train doctoral students, which I have in my lab, and I'm very fortunate for them. All right, so although my talk title says Repurposing Drugs for Osteosarcoma, I'm only going to talk about one drug, which is Riluzole. So here's some background on Riluzole, on glutamate. So I'm going to discuss briefly about the receptors being expressed in cancer, and then move on to the findings from my lab. So I have basically 4 small themes that I'm going to talk about. I don't want to go through the list. Basically, it's first testing Riluzole in vitro, then delivering Riluzole through nanoparticles. Then we looked at the mechanism of action of Riluzole in osteosarcoma. And then we have the latest project, which is efficacy of Riluzole in osteosarcoma cell lines and patient-derived samples. And that's the work that is currently unpublished. And those slides will not be in the recording because in the online version, because they'll be edited out, just a heads up. So glutamate, as we all know, is an amino acid. It's actually a non-essential amino acid, and it's a major neurotransmitter an excitatory neurotransmitter in both central nervous system and the peripheral nervous system, and plays a very important role in memory and learning. I just want to grab my laser pointer. All right. Very, very important for memory and learning. That's in neuroscience. However, in other cells that are rapidly dividing, it's a bioenergetic substrate, which means the cells use this as a food, a source. And more importantly, glutamate is also a signaling molecule, and this is seen in peripheral tissues such as testis, pancreas, pineal, pituitary, and adrenal glands, and more importantly, in bone development. So amyotrophic lateral sclerosis disease got popularized because Lou Gehrig, a famous baseball player, actually got this disease. And in the US, it's popularly known as Lou Gehrig's disease. And what happens in this disease, it's actually a progressive neurodegenerative disease and pretty rapidly progresses. And in 30% of patients, the glutamate levels basically get enhanced in the cerebrospinal fluid. And this disease became popular when Lou Gehrig announced his retirement when he was diagnosed at the age of 35, 36 of his retirement at the peak of his career. And that's basically the relevance to ALS and the Riluzole and glutamate levels, which I will return back to as I speak. So in a glutamatergic system that exists in the nervous system, and I'm showing you a synapse and the accessory cells from the nervous system only because these receptors and transporters have been extensively studied in the brain. So number one, glutamate is released by 2 processes. One is the vesicular transport, as you can see here. And the other one is, I'm going to move this so I can, the other one is called the XCT, which is an antiporter for cysteine and glutamate. So it basically imports cysteine into the cell and exports glutamate. So that's how glutamate becomes available either via the excretion through the vesicular transport or via the XCT. And once it is released in the synaptic cleft or in the extracellular space around the cells, it can bind to two different types of receptors. The first one is called the ionotropic receptor that I listed here. And there are three different types: the AMPA receptor, the NMDA receptor, and the KNET receptor. And these are nothing but ion channels that the ions pass through. The second category is the metabotropic glutamate receptors, which are shown here on this cell, which act as a receptor. However, the one that are located here may have inhibitory function. So I will show you in a minute the different types of metabotropic glutamate receptors. These are nothing but cyclic GMP-dependent receptors. So here we have another system, which is the EAT, which is the excitatory amino acid transporter. So here they are, the three different types. So what happens is these are the transporters that transport the glutamate back from the extracellular space into the cell. So they are basically utilized for reuptake to maintain the levels of glutamate in the synapse so that the cells don't get overexcited. So what happens in neurodegenerative diseases is that excessive glutamate signaling, because of excessive release and no proper reuptake, can actually lead to many other disorders such as Alzheimer's, Parkinson's, besides ALS. And this is also seen in brain injury. In tumorigenesis, however, AMPA receptor, NMDA receptors, glutamate receptors are shown to participate, and I'm going to show you a list of different cancer types where these receptors may be involved. The XCT, which I showed you as a protein transporter, antiporter, basically brings in cysteine and exports glutamate, can actually be elevated, and it's been shown that the XCT over expression can actually be oncogenic. And I'll explain a little more later. So when we look at the metabotropic glutamate receptors, there are three different types. Group 1 is typically what is seen in cancer cells, because these, as shown in the neuro system, that they are actually excitatory, meaning that they actually stimulatory because they use molecules such as IP3 and DAG, and they activate different pathways, which I'm going to show in the next slide. By IP3 and DAG and activating many other cell signaling pathways, such as the PI3-AKT pathway, the NF-kappa B pathway, for enhancing growth and survival genes to survive, for proliferation of the cells, as well as inhibiting apoptosis, basically to enhance the progression of cancer growth. Now, mGluR1 was also shown, which is basically a part of the metabotropic glutamate receptors, can also influence the activity of glutaminase, which converts glutamine into glutamate. And this glutamate gets secreted out through the antiporter XCT. And this molecule then acts on the receptors, basically helping in the exocrine, helping in the autocrine paracrine function. Other pathways are also activated by this, which are involved in metastatic properties and cell survival and growth. So this is just a generic, signaling mechanism. I'm not focusing on the ionotropic receptors because my research focused on the metabotropic. So I'm actually going to just show you the metabotropic glutamate receptors. And here you see a long list of different types of cancers, where you see different types of mGluR receptors have been shown to be expressed, which contribute to the malignant behavior of the tumors. And in osteosarcoma, we and other labs have shown that mGluR4 and mGluR5 are expressed in most of the osteosarcoma cell lines. So about 5.5 decades after Lou Gehrig's death, Riluzole was approved by FDA as a drug to treat ALS, and it's nothing but benzothiazole drug, and its complex structure is shown here. What it does is actually reduces glutamate levels by blocking glutamate release in the cells, and it also enhances the glutamate clearance. And it does that by actually increasing the activity of the EAAT transporter. Now, as I said, Riluzole, that glutamate secretion is excessive glutamate stimulation is seen in many psychiatric and neurodegenerative disorders. Riluzole has been actually tested, and for some of these diseases, under clinical trials for that. Riluzole is available as tablets, 50 milligram dosage. It's also available as liquid for people who have swallowing issues. And more recently, in 2021, Riluzole was approved as in a form of a film, so that it's easy for patients to ingest Riluzole. Now, why repurpose any drug? Because of fiscal benefits. Here are the data that was published in JAMA Health Forum. You see that for a new drug to develop, the expenses are anywhere from 1.4 to 3 billion per new drug approved, as opposed to 52 to 81 million for a new drug that is repurposed for drug approval for any other disease. Besides the fiscal gain, it also takes a longer time for an average new drug to be developed for cancer therapy, 10 to 17 years, and for repurposing, usually shorter, 3 to 12 years. And more importantly, less than 1% of new drugs enter clinical trials, so that's actually a huge loss of time in terms of drug development. So here, this is the scheme, how one basically goes about repurposing drugs that are used for non-oncology purposes. And here is a list. And then you use any of these drugs, find a target, and then you do in vitro experiments. Then you perform in vivo experiments after you get supporting data. If this is looking good, then you go forward with clinical trials, and then you purpose the drug for cancer therapy. So why Riluzole? Because Riluzole, as I had already mentioned, it was approved as a drug in 1995 for ALS treatment. So there's enormous amount of data on toxicity and tolerance and pharmacokinetics, and it's continued to be monitored for pharmacokinetics and bioavailability, especially in this article that I'm showing here, where oral film was approved for usage by ALS patients. So it continues to add on, so we can use the data and use it for repurposing Riluzole for cancer. When we started this work in about 2014 or so, 2015, this is what we knew about glutamatergic system in osteosarcoma, is that the system was shown in cells called MG63 cells, which are osteoblast-like cells. And these cells were shown to spontaneously release glutamate, and the glutamate release was blocked by Riluzole. So there was not much known beyond that. And then the paper also said that they were like some apoptotic-like features in the cell. So that was all. And then later in 2013, a genome-wide association study of people of European descent, in patients of European descent, it was shown that mGluR4 is expressed highly compared to normal subjects. And here is a picture of SALS2 cells, which are osteoblast-like cells. You see the NMDA receptor. This is actually the ionotropic receptor being stained with an antibody, and it shows that these cells actually express the glutamate receptors, the ionotropic one, specifically the NMDA receptor. Now I'm going to move on to the findings from my lab. I'm going to start with the effect of Riluzole on osteosarcoma cultures in vitro. For this study, we used 2 cell lines, mouse OS482 cell line, and then LM7, which is a human cell line. Both these cell lines are metastatic osteosarcoma cell lines. And most of the data I'm going to show you is on LM7. So here is LM7 data. The first thing we checked was glutamate secretion, and here is a glioblastoma cell line, the U87, that secretes high levels of glutamate. And compared to that, LM7 still shows, LM7 secretes glutamate to significant levels. And here is our negative control. And we performed this assay in media containing glutamax so that there's no interference of glutamine that's been oxidized. So the next question is, what happens when we block glutamate secretion in these cells? Well, so here is a staining, immunocytochemistry staining, and here is just DAPI staining the nuclei, and here is basically what we call as a proliferative assay, where we are staining the cells with Ki-67 in the presence of different concentrations of Riluzole. So what we see is that as the dosage increases, the number of green cells decrease, which means that the proliferating cells decrease. However, you can also see that the blue bars decrease, which means the total number of cells are also decreasing. Well, this is because the cells are actually dying, and we see that in an apoptosis assay. Here is just all total nuclei, and as you increase the dosage, the number of tunnel-positive nuclei, which basically scores apoptotic cells that are dying due to the presence of Riluzole, are seen, and you can see the increase in the percent apoptosis. Riluzole also prevented migration compared to control. Here is a scratch assay where you actually measure, you make a scratch and you measure the scratch in the presence or absence of Riluzole, and you can see that the cells do not close the gap. So you can see that tabulated here, where the control cells basically close the gap and the Riluzole don't, because they don't migrate. So both graphs basically show that. What we wanted to know was what receptors are being utilized that are targeted in osteosarcoma for growth that are targeted by Riluzole. And when we tested two separate inhibitors targeting both mGluR1, which is the CCPD, or the mGluR5 inhibitor, which is Phenobam, what we found is that Phenobam was basically replicating the data we see with Riluzole, which is when you have Phenobam or Phenobam with DHPG, which is a stimulant for both the mGluR1 and mGluR5, you can see that Phenobam can actually prevent proliferation just as Riluzole in LM7 cells. Similar to Riluzole, it also increased apoptosis, whether alone by itself or even in presence of DHPG. And here we show that LM7 cells do in fact express mGluR5 receptor. And here is a positive control, which is the glioblastoma cell line, UH7. So when we knock down the GluR5 expression from the LM7 cells, here is the wild-type LM7 cells, here is the control shRNA, and when we knock down using a specific shRNA for mGluR5, we see that the proliferation, or in this case, the colony-forming ability of LM7 cells is lost when you knock down mGluR5, and you can see here, and here is a knockdown showing, Western blot showing that mGluR5 is basically not expressed in the knockdown. So to summarize, Riluzole blocks glutamate-dependent signaling and cell proliferation. It induces apoptosis in osteosarcoma cells in vitro, and we show that mGluR5 is required for osteosarcoma cell proliferation and colony formation. Then we asked, how can we enhance the efficacy of Riluzole? And in collaboration with a chemistry professor at Hunter College, we conducted this study, Hiroshi Matsui. So I'm going to show you both in vitro and in vivo data with using iron oxide nanoparticles. So here basically are the particles that we use. This is the transmission electron microscopy images, and here is an enlarged image of the single cage. I mean, 3 cages here, 3 or 4 cages. This is basically a rendering of the cage. You can see it is hollow. So Riluzole can be filled. Each cage is 15 to 20 nanometers in diameter, in size. And Riluzole is 76.4 angstrom. So when we measured after loading the nanoparticles, I will call this nano cage because it's like a cage, and we'll use another particle in the study, which I will refer to as nanosphere. However, so when we load the nano cages with Riluzole, we estimated that about 50 Riluzole molecules are loaded in each nanoparticle. So after you load the nanoparticles, you coat the nanoparticles with DHCA dextran capping. DHCA is dihydrocaffeic acid, and which is coupled to dextran. And you cap this iron oxide nano cages after loading, and then they can be applied to the cells. And here is the data with cultures. So when we added, these are the controls. So we have just control DMSO And here is the neat nanocages, and here is a neat nanosphere. Nanosphere is just a spherical iron oxide structure, which is not hollow but solid. So when you load it over the sphere, and then you cap it with the reagents dextran, DHCA, and dextran. And here is Riluzole, neat Riluzole, which is able to, of course, reduce proliferation and increase apoptosis. However, when you compare all of these samples, the most apoptotic, highest percentage of apoptosis is seen when Riluzole is delivered with a nanocage, which you can see here when you look at the percent apoptosis. We then conducted the in vivo study where we used NSG mice. We used LM7 cells that express firefly luciferase. After injecting the cells subcutaneously, we divided them into 6 groups, actually. So there are 3 treatment groups and 3 control groups. The 3 control groups were injected with PBS, ionosphere, or inocage, and the treatment groups either had free Riluzole or Riluzole loaded in a sphere or Riluzole loaded in a nanocage. Here is the data from manual measurements using vernier calipers. These are the control samples from all the 3 different control groups. And here is the treated samples. You can see that the lowest tumor size, actually tumor volume, is seen in the group that had in which the Riluzole was delivered using nanoparticle. And here you can see the tumor volume percentage between the 3 groups, where you see the smallest size with the Riluzole delivered via nanocage group. Here is a bioluminescence data, and here is an image of the control mice. This is just one of the images of all the mice that were, and I wanted to bring your attention to the scale here, which is 6 times higher than this scale. And here are the treated groups, where you can see that the Riluzole, compared to Riluzole, the nanosphere delivered Riluzole shows reduced tumor size, and it gets further reduced in the case of mice which have actually Riluzole delivered through the nanocage. So you can see the difference here. So what it tells you, okay, so then what we did is took the tumors out, and then we stained them with DAPI, and here's the H&E stain. We also looked at the apoptotic cells. You see that all the treated groups, Riluzole or Riluzole delivered through nanosphere or the nanocage, show increased apoptosis. And when we look at the measurements, here are all the samples. I want to focus on the treatment groups. You see that the highest percentage of apoptosis was seen in the animals in which Riluzole was delivered through nanocages. So to summarize this, what we found was that Riluzole was most effective when we delivered it through nanocage compared to free Riluzole or Riluzole delivered through nanosphere in reducing tumor size, as well as in inducing apoptosis both in vitro and in vivo. So for the mechanism of action of Riluzole, so I'm a molecular biologist and I love pathways and molecular mechanisms. So we had to do this. How does Riluzole work in this cell? How does it bring about apoptosis? So we use LM7 cells as this model. And before I go into details of the mechanism, I'm not actually showing any data because of the lack of time. We have limited time. So I'm just going to show you the model that we came up with after we presented our results. I just need to give you a little bit of background on associated protein, also called YAP. It's a transcription factor. It's a downstream regulator of Hippo pathway, which controls proliferation and apoptosis. The oncogenic proliferative property of YAP depends on its interaction with TAA domain containing transcription factors. And YAP is shown to be overexpressed in many types of cancers, including osteosarcoma, and is also correlated to low survival outcome. When you use inhibitors of YAP, which is waterporphin or CA3, it has been shown to reduce primary tumor growth. And when YAP is knocked down in osteosarcoma, it results in tumor shrinkage. So that's the background for YAP. So here is a model that we came up with. We actually use models where we, as I go through, I will explain more. So bottom line is that osteosarcoma cells, when treated with Riluzole, undergo apoptosis. what happens is that riluzole increases ROS, which is the reactive oxygen species in the cells. If you recall, I said that riluzole, I didn't say that yet. So XCT is one of the transporters that brings in cysteine and exports glutamate. So what happens is that if the cysteine is not imported into the cell, it can increase ROS because glutathione is not produced. I will show you that slide again. Anyhow, so ROS is produced, which basically activates c-Abl kinase. c-Abl kinase phosphorylates YAP at a specific residue, which is the tyrosine 357. And once YAP becomes phosphorylated at this residue, it basically develops affinity towards P73, which is stabilized under conditions of DNA damage. And they both together and with other accessory factors bind to the promoter of pro-apoptotic genes, and that's how apoptosis is executed. So I actually summed up a lot of information here, but the bottom line is we basically knock down the cells of c-Abl and YAP and proved that this is what goes on in these cells. And we are actually working on this mechanism further, and I will provide more data in the future. So this is here is an example of all of the different cancer types where riluzole has been tried as an anti-cancer drug. The list is really long, and we are the ones focusing on osteosarcoma. I hope more people join us in doing so. And there have also been clinical trials. A lot of work with riluzole has been done in melanoma, and when I started, this is what I basically followed the literature on riluzole, the use in melanoma and the mechanism, etc. And I know the PI that works at Rutgers, so it's good to have someone doing the work, and then you follow what they do. And but the types of cancers are different, the mechanisms are different. However, it's a good support system. However, okay, so more recently, colorectal cancer. In colorectal cancer, riluzole has been tried in phase 1 in combination with 2 other drugs. And in solid tumors, 2 phase 1 studies have been conducted, one in combination with sorafenib, and the other one, the more recent study, this was in 2022, where prodrug of riluzole was used. And the reason for using prodrug is because riluzole is metabolized differently in human individuals because of the heterogeneity in the enzyme that metabolizes it. So if you use a prodrug, you basically bypass that issue. And this was a dose escalation study where 420 milligrams was used, and they looked at the side effects in the patients. What I want to point out basically is the dosage also that was used in all of these studies was anywhere from 150 to 100 milligrams twice a day. And the maximum dose, the MTD for prodrug, which is riluzole, was estimated to be 420 milligrams per day. Yeah, here is the ROS story. I am sorry, I spoke a little bit about this before. So here, what riluzole does, this is not just from our lab, but from the work of other labs that have done this, this is just a model to show where all riluzole actually acts on. So here, it can block the AMPA receptor, the NMDA receptor, and the mGluRs, which is the metabotropic glutamate receptors. So that can actually prevent the cell proliferation in the cancer cells. What it also does is block the XCT, which helps in secreting glutamate. So if you block that, what happens is the import of cysteine is also affected, because of which glutathione synthesis is blocked. And when that happens, ROS increases. In general, an ROS is very damaging to the cells because it activates many different processes leading to apoptosis. Generally, cancer cells operate at very high ROS level, and when you further perturb it, it actually becomes detrimental to the cell. So that's how the cells basically undergo apoptosis. And riluzole has been shown in neurons and other associated neuron-associated cells that EAAT is actually, the activity of this is enhanced by riluzole, which means that this actually helps in pumping the glutamate into the cell. So that's another way it can actually prevent the autocrine signaling. Now here is a study. The next few slides are going to be edited from the recording because they contain unpublished data. And I'm going to basically discuss this data where we use 11 cell lines, osteosarcoma cell lines, and 4 patient-derived cell lines. And we're looking at the efficacy of riluzole in these cell lines. So here we have, there are 3 different pairs of cell lines, non-metastatic and metastatic. So you have SOUS2 and SOUS2 LM7, MG63, MG63.2, that's non-metastatic and metastatic, HOS, MNNG, HOS, MNNG, and 143B. First one is non-metastatic, and these 2 are metastatic. Basically, these are the daughter cells of that. We also have U2OS cells, which are popularly used in osteosarcoma research. We have 2 mouse cell lines, the K7M2 and OS482. And then we have also included a fetal osteoblast cell line that has been immortalized. Then we've used 4 patient-derived cell lines, which were generated in Dr. Healy's lab by Raj. And these are OS24, 29, 33, and 69. And all of these carry different types of mutations in them. One of these PDX was isolated from a metastatic site, and the 3 of them were from the primary tumors. So we tested to see if these cells secrete glutamate, and all of them, the primary tumors that we have, I've listed there, with respect to FOB, which is, so basically all the values are in relative to FOB, the secretion of glutamate in FOB. So you see they all secrete glutamate increasing, from day 0 to day 7. Same as the metastatic cell line. In general, metastatic cell lines tend to secrete more glutamate. And that may also be because they proliferate much faster, so it's more cells contributing to it. And the PDX cell lines also secreted glutamate into the media. Only one of the exceptions is the OS24, which secreted less than FOB. So otherwise, other ones, even OS69, so more or less they are. So OS69 secretes similar to FOB, but OS24 secretes a little less than HFOB. In a cytotoxicity assay, or growth inhibition assay, I should say. This is an MTT assay where we basically tested all of these cell lines. We see that all of these cell lines show dose-dependent increase in inhibition when riluzole was added. And so does the PDX cell lines also show inhibition. And point to note is that although they all show dose-dependent inhibition in growth, in the presence of riluzole, the IC50 value may vary from cell line to cell line. And then because we had, we knew that riluzole may act through increase in ROS, but this is a general phenomenon for all of the cell lines. So we tested all of the cell lines and measured ROS in these using a fluorescent assay. So you can see that these are we have used H2O2 as the positive control. The red and the green bars are the ones that show dose-dependent increase in ROS in all of the cell lines. So SOUS2 LM7, U2OS, all of these cell lines show dose-dependent increase. And here are the PDXs that show the same. However, OS69 showed increase only at 50 micromolar and not at 100 micromolar, and we don't know yet why, but we will figure that out. Point to note here is that 143B was the only cell line that did not show increase in the ROS, and that could be because it harbors a KRAS mutation, which was overexpressed in these cells, and those are expressed at very high levels. And so that actually somehow helps the cells to combat increasing ROS. So it basically resists ROS increase, and we are working on this. We have data that I'm not showing today and sometime in the future. All right, so the next assay we performed was the Boyden chamber invasion assay, where we try to test the invasive ability of the cells in the presence of riluzole, and all of our data shows. So here we have different cell lines, the MG63 and 63, SOUS2 and LM7, non-metastatic, metastatic, non-metastatic, and metastatic. You see that the presence of riluzole decreases the ability significantly. Decreases the invasive ability of these cells significantly. Same is true for U2OS, HOS, MNNG, and 143B. You can see that compared to the control, riluzole decreases the invasive ability of the cells. This is also true for the patient-derived samples. We can see that, yes, compared to the TMSO control, which is at the center, I should explain that the leftmost corner, the negative control, is basically cells grown in no serum at all. I mean, very low serum so that they don't grow. It's just a negative control. And then here you have cells growing with the vehicle, DMSO, and here are the cells that are grown in presence of Riluzole. And we grow them only for 18 hours on the membrane so that, we can measure the mobility of the cells or invasive ability of the cells without, before they die, basically. I mean, because Riluzole does kill them. So we try to use concentration of Riluzole that is not very high. So we're using 25 micromolar Riluzole here. And here is the data. So basically, to summarize, osteosarcoma cells, all of the ones that we have tested, 11 cell lines and 4 patient-derived samples, all of them secrete glutamate, and Riluzole blocks growth in osteosarcoma cell lines and PDX cell lines when you prevent the glutamate secretion. And we also shown that Riluzole prevents invasive ability of osteosarcoma cells and the patient-derived samples. So here is the the cycle or the steps to repurposing a drug, and we are smack in the middle of in vitro experiments and in vivo experiments. We've completed in vitro experiments and the data that I just showed you, some of it, and we have more data that basically looks at this the V-glut, which helps in the vesicular transport of glutamate into the extracellular space, and the XCT measured all of that. I didn't show that data, but the point is that that data is the manuscript is in review right now, and we are about to begin our in vivo experiments. And thanks to MIB Agents for the Osteosarcoma Award, that we will be using those funds to conduct the in vivo experiments. I'd like to thank, of course, my students who work in my lab, Saida, Mariam Azim, Okanjang, Pooja Rao, and Shraddha Chantakuri. I didn't show any data from Pooja's work. However, she has very interesting data that I will be hopefully sharing with you all. And here are all the former students, my undergrad students who helped me with this work. Big thanks to Dr. Healy, who has provided us the PDXs and also invaluable support and guidance in the project. And thanks to Raj, who basically made this, isolated the PDXs and grown them and shared with us. And I'd like to acknowledge Hiroshi Matsui from Chemistry Department at Hunter College, who has helped us with the NanoCage work. And big thanks to all the funding agencies from the NIH, as well as the internal grants at CUNY and also the MIB agents. Thank you.
Thanks so much, Dr. Mahajan. And amazing that you were able to kind of get through all that pretty clearly in such
I hope I didn't rush through, so you can ask me questions to clarify.
Yeah, well, we definitely have some questions, and I guess I'll hand it over to Walker to kick off with some of the questions.
We'll just stop sharing now.
Sure.
The first question that came in is a two-part question. The first part asked, does LM7 cells spread to the lung spontaneously? If so, does your therapy prevent lung mets?
Great question. So yes, they spontaneously metastasize, and they're very, very aggressive. We tried actually a model with the metastatic model, which is a dirty model where you inject in tail vein. We haven't published the data, and we tried prodrugs with that. So we're still analyzing the data. Bottom line is, yes, it prevents to some extent, and how much is what we need to figure out. But it is, yeah. But I'd say that's not the greatest model to use because their cells are so aggressive, but they do spontaneously metastasize to lungs.
Gotcha. And you may have already kind of answered this second question, but the second question from the same person asked, does LM7 cells work through tail vein assay? Does your therapy prevent IV lung metastases, which I assume is in vitro lung metastases?
That's the same question as the first one, I'm thinking. Is that right?
Yeah, I think they're pretty similar. Technically,
what was the first part of the question? Sorry.
The first part of the question, do LM7 cells work through tail vein assay?
That's what so we did the tail vein assay, and we would want to use a better model for our in vivo studies. So that's what we hope to do. Yeah.
Okay. Yeah. Thank you.
You're welcome.
Sona, do you want to take the next question?
Yeah. Thanks. So the next question was, how did the IC50 values of the cell lines compare to riluzole concentration in humans?
Great question. So I'd say that some cell lines are more sensitive than the others to riluzole, and that probably depends on, like for example, the 143B is the best example I can give you, is that the IC50 value for that is the highest. So 100 micromolar is IC50, which is terribly high, and that is due to KRAS mutation. And what we found is that when we try to when we actually combine a KRAS inhibitor with riluzole, it does wonders. So in cases where riluzole, where cell lines or let's say patient samples have a high IC50, it may need to be combined with other drug. And the good news is that riluzole does work well with other agents. All right, so the cells are suspended. So this is a double chamber, top chamber and a bottom chamber. The cells are suspended on the top chamber, and then here you have the media. This is where you add the drug. You grow the cells 2 to 24 hours. We grew them for 18 hours. And then what happens is, because of the invasive ability, you're actually using a membrane. The green part is basically the membrane with a certain porosity, so that the cells actually have to squeeze in and go through that. And so some of the cells, depending on the invasive ability, will be seen on the other side of the membrane, which is the reverse side. So you take this membrane and you stain them, and you can see the difference in staining. So when you look at so you see that this is a negative control, and here is a positive control where cells were able to cross through that, membrane, and they lodge on the other side of the membrane. So basically, and when you add riluzole, you can see that it prevents the invasion of the cells to the other side. I hope I answered that question. It's called the Boyden chamber assay.
I think there's a follow-up question to that. Just where did you put the drug?
Oh, the drug is in the bottom chamber. Yeah, so basically, because of the presence of the drug, they don't cross the membrane. They just stay on top.
Thanks. And then
Good
Simon, I think
assay.
you had a question.
Yeah,
So-
I was curious if you could talk a little bit more about why the nanocage particles are advantageous over the nanosphere.
I think it's just the release of riluzole, probably. So when we did the in vivo work, I think that the delivery of riluzole, when you put it in the nano cage, takes a little more time as compared to free riluzole, which is available almost immediately. But with when it is in the cage, it takes time for the release, and so the effect is longer. It's longer acting, even though the drug is the same. Yeah, it's a great model, and we love the results, except that it's not so practical in human use, only because the particles would accumulate over time, right? And it could lead to iron toxicity, because these are iron oxide particles, even though they're smaller. And they were able to penetrate through into the capillaries, into the tumor. We actually looked at that, because the vasculature in the tumor is more, the porosity is different, so that's why it would get in. Yeah, but the only issue is that it would not be more useful. So what we are hoping to do would be more work with the prodrugs that will prolong the bioavailability of the drugs in the system. Yeah, so the release is through what is called as the Brown. We actually had a follow-up paper, not myself, but the chemistry professor had a follow-up paper where he explains the physical dynamics of how the drug is released. The particles go through what is called as Brownian motion, and when you want to release this in the animals, you can actually apply magnetic field around the animals, and then the particles spin to release the drug. Yeah, so yeah, and they are basically taken up through the endocytosis process.
Thank you. That's
Thank you.
the nanocage particles sound really cool, because I've only heard of the spherical ones.
Yeah, these are, yeah, and they're cool because they are like a hollow, so you can fill them with any drug. In fact, we use them. I didn't show the data today. We published a paper where we use these nano cages to deliver, siRNA, which are small, short sequences of RNA that are inhibitory, that block the expression of certain genes because you choose sequence specific. Yeah. So basically, we were able to use siRNA and deliver it using the nano cage. And we had to apply in that case, because in the case of Riluzole, it's more or less spontaneously released in the culture or in the animals. But to release the DNA or the RNA, we had to apply magnetic field, so the magnetic particles spin and then they release the RNA or the DNA nucleotides, oligos, basically.
Dr. Mahajan, I had a question about, and I might have missed this when you were talking about Riluzole. So I know you had a slide where you'd shown that the mGlur 4 and 5 were overexpressed in osteo. And then I did catch another one where I think it was Phenobomb that targeted mGlur 5. And I was
Yes.
wondering, for Riluzole, is that focused more on mGlur 4, or does it target 4 and 5 equally?
So the great question. So Riluzole works on all channels only because it doesn't work directly on them. It blocks glutamate. So by blocking glutamate, it's taking away the ligand which binds to this receptor. So it generally blocks all the glutamate dependent signaling. And it also blocks the XCT, which brings in cysteine. So it increases the reactive oxygen species. So the bottom line is Riluzole acts by working on many different receptors at the same time. And Riluzole has been shown to do much more. We actually, if I could find that again, sorry. Another, yeah, this one I want to show you. Another slide, if I can, here. So this was, we published this review in 2021 when we were quarantining from COVID and all that. So this is the mechanism of action of Riluzole. So it tends to work on all of the different channels, the ionotropic, which are the channels for calcium and sodium, the XCT, which I showed you, the mGlur, and the glucose uptake, etc. So it works on multiple channels, and here are all the different pathways that it targets. So bottom line is that regardless of what it targets, it tends to actually induce apoptosis or cell cycle arrest, etc. in different cancer types. This is basically a comprehensive review of action of Riluzole in different cancers. I'm going to stop sharing again, and yeah, I hope that answers the question. It not completely, I know I completely answered that, but oops. Sorry.
No, I was trying to do like kind of a quick, like a little looking at the Phenobomb, which I think you had mentioned.
Yes. So the Phenobomb, yeah, I'm sorry, I think I probably answered tangentially. So Phenobomb specifically targets the mGlur 5, and Riluzole can block more than mGlur 5. That's why we needed to figure out what is it blocking in this cell. And when we tested all these cell lines that I showed you in the last study, we actually looked at mGlur 1 and mGlur 5 expression, and they all do express at different, varying degree. They all express either the vesicular transporter, or some of them have higher levels of XCT, and they express either mGlur 1 or mGlur 5. Yes, so Riluzole could be blocking either of those, or both in some cells.
And related to that, when you were showing some of the work on, I can't remember if these were PDX or cell lines, but there was one, I think it was like OS69 that did not have an increase in ROS. And then like before that, there was like another cell line that had a lower expression of glutamate. I think it was like OS24.
Yes.
And so curious, like, is there a way to, is there some sort of biomarker where you could kind of determine which types of disease would kind of respond to Riluzole best?
That's a great question, because we in fact have a protein that one of my students, Mariam, is looking at. So it's called EGR, and she had presented at Factor 2023, I believe. So when you treat with Riluzole, EGR generally goes up in pretty much most of the cell lines. So it, in some cell lines, so it's usually suppressed in metastatic cell lines. But when you treat with Riluzole or any other chemotherapeutic agent, the levels of EGR1 go up. So that we're hoping that we can use as a treatment biomarker for osteosarcoma. Because it's usually suppressed, but when you add, but in some cell lines, the basal levels are a little higher. So we need to select a bar beyond which or above which we have to say, okay, this is positive and this is negative. In terms of whether or not it's elevated in response to Riluzole.
And maybe I'll see you one more question before we have to wrap up, which is, I know you're that was actually helpful to kind of see where you are in the process and you're moving to in vivo models. But I was curious, I was kind of looking to see whether or not the drug has ever been used in dogs. And if you have, it looks like it has been tested in dogs for other, indications, but as you know, dogs commonly get osteosarcoma, and could be an interesting.
Yeah, actually, Dr. Healy had suggested we do this in dogs. It's just that everything takes time and energy, and we haven't gotten there yet. So Riluzole has been tested in dogs just for toxicity purposes. I think there is a paper that says Riluzole in dogs, so basically for pharmacokinetics and toxicity studies, they did a small study in dogs. So it has been tested, but not against cancer per se or osteosarcoma per se. But that's something hopefully we can do.
Yeah, and sorry, I lied about last question. One more question. As I
No, please go ahead.
I was just thinking about, like clinical translation and kind of where this potentially could fit in clinical treatment. So, if it were to move forward, kind of where do you see it fitting in clinical treatment?
So it's interesting you asked me the question because I've been thinking about it a lot after I saw talk on osteobites last time, Mecacron, and he listed a lot of different reagents or other drugs that can be used, or MABs that can be used against osteosarcoma. And I was like, wow, this is alarming, because for one patient, if you use all these drugs, we're surely not doing a good job. But something like Riluzole, which can target a variety of different types of cells with differences in the genotype, because of there's so much heterogeneity, could be used as something that can be used in addition to other drugs, I think, because I don't know, single agent therapy is just too, it doesn't fulfill the promise. So I think that it can be it can be used as a supplemental drug, but we just have to see like how this goes though. But I'm hopeful that this will be useful in one form or the other. And we have another study. I didn't show Pooja's data, but that's basically Riluzole has been tested as a radiosensitizer also, and it seemed to work really well in osteosarcoma, and at least in what we are testing. And so we are very, very hopeful that in one form or another it will be useful to the patient community soon.
Yeah. And it was, your slide about kind of why repurpose and kind of the time and the money, I think was really compelling.
Thank you.
Yeah. Thank you for sharing that. Thank you so much, Dr. Mahajan, for joining us on Osteobites today and for making it better for osteosarcoma patients. More information on this and all osteobites can be found on our website at mibagents.org, on our MIB Agents YouTube channel, and at your favorite podcast place. And we hope to see you next week on November 7th. We will be joined by Dr. Arianexis Aquino-Lopez. She is a physician scientist from Texas Children's Hospital and the Baylor College of Medicine. And she's also one of our 2024 OutSmarting Osteosarcoma Young Investigator grant recipients. And she will provide an overview of her funded work on repurposing virus-specific T cells as an immune therapy for osteosarcoma. You can find our Osteobites lineup for the next few months on our website. And if you have any ideas for topics that you'd like to hear about, please feel free to email us at events@mibagents.org. Thank you again, Dr. Mahajan, Walker, and Sona for spending an hour with us today. And thanks to all of you as well for joining us on Osteobites. We hope to see you back here next week on the 7th when we chat with Dr. Aquino-Lopez. Thanks, everyone. Thanks so much, Dr. Mahajan. Thanks, Walker.